Hello everyone, and welcome to this video on the special senses. Today we're going to start with the sense of smell, and we're going to start with a video.
Olfaction, the sense of smell, is far more complex than gustation. Most people can detect between 2,000 and 4,000 different odors. Olfactory receptors are located in two small areas, on the roof of each nasal cavity, directly below the eyes. Each olfactory neuron has 10 to 20 hair-like structures called cilia, which give the olfactory epithelium more surface area with which to detect smells. Amazingly, the exposed surface area provided by those cilia of the olfactory cells is almost equal to the entire body surface area, about 2 square meters. The olfactory cilia are typically covered by a coat of mucus. When you inhale a substance in the air that causes an odor, it dissolves in the mucus and binds to receptors on the cilia. Because the olfactory epithelium is in the upper part of the nose, it may not always be the best place for detecting odors. In normal relaxed inhalation, only about 2% of the air we breathe is actually carried to these olfactory regions. That's why we sniff. When we smell something, it draws air upward across the olfactory region. Olfactory nerve fibers pass through openings in the skull and enter a pair of olfactory bulbs in the middle lobes of the brain. There, the nerves connect with neurons called mitral cells, with structures called glomeruli. Each glomerulus receives only one type of odor signal. The signals are then sent to two main destinations. Some go through the thalamus to the cerebral cortex, where smells are consciously interpreted and identified. Olfactory signals are also sent to other regions of the brain, including the limbic system, where the odors may cause specific emotional responses and memories. For example, odors associated with dangerous or unpleasant things, like a skunk, can trigger a fight or flight response.
Okay, so a couple questions after watching that video. First, how many different odors can we smell? That video said 2 to 4,000, although new research suggests it may be closer to 1 trillion, so it's a lot. Another question, what is the surface area of the olfactory cilia? And it's about equal to the entire skin surface, which is pretty amazing considering that it's in a tiny piece of your nose. To what 3 places in the brain does olfactory information go? Signals are sent down the olfactory tract to the thalamus, the hypothalamus, and the limbic system. From the thalamus, it's going to go to the cerebral cortex, and then we're going to consciously interpret it as smell. For the hypothalamus and limbic system, it's more going into our emotional response. And describe the olfactory pathway to the cerebrum. This figure will help. So signals go through these olfactory receptor cells to the olfactory bulb, and then back through the olfactory tract to the thalamus, hypothalamus, and limbic system. Then our next question is describe transduction in the olfactory system, and this figure sums it up pretty well. When an odor here binds to its receptor, it activates this enzyme called adenylyl cyclase. Adenylyl cyclase then converts ATP into cyclic AMP. Cyclic AMP then is going to open the sodium channel. That's going to allow sodium ions to enter, which leads to depolarization of the cell. And if the cell reaches threshold, an action potential will be generated. Then next we're going to talk about the sense of taste. this video.
Gustation, the sense of taste, is important because it provides information about the quality of the food we consume. In particular, it helps us determine whether what we put in our mouths could be harmful to us and shouldn't be swallowed. The tongue has folds and bumps on its surface called lingual papillae. These peg-like projections give the tongue a slightly rough feel. The receptor organs for gustation are called taste buds. We each have about 10,000 taste buds, most of which are located in the lingual papillae. A taste bud is actually a cluster of about 100 taste receptor cells. During chewing, water-soluble substances from food dissolve in saliva and enter a taste pore of the cell, which then synapses a nerve fiber at its base, sending taste information to the brain.
There are only a few basic tastes: sweet, sour, salty, bitter, and a fifth one called umami. Umami is a Japanese word which means delicious. Umami is a pleasant taste brought about by amino acids like glutamate that enhance other taste qualities. That's why monosodium glutamate, or MSG, is sometimes used as a food additive.
Taste and smell both contribute to our perception of flavor. In fact, smell plays the greater role.
Okay. So gustation is taste, and it also uses chemoreceptors, which are found in the taste buds. The bumps we see on the surface of the tongue are called papilla, and the taste buds we'll see are inside of the papilla. So we're going to talk about transduction for the different tastes. Salt and sour are pretty similar. First of all, we're most sensitive to bitter substances and sour substances because they may be harmful to us. And a sour taste happens when hydrogen ions open ion channels in gustatory receptor cells, such as we see right here. A salty taste happens when sodium binds to, it opens ion channels in gustatory receptor cells. So sour is due to hydrogen ions, salty is due to sodium ions. And then for sweet, bitter, or umami, these chemicals bind with the receptor, which activates a second messenger, which is then going to lead to depolarization of the receptor cell. And olfactory receptors contribute about 70% of our sense of taste. That's why you can't smell when you have a cold. Other things that contribute to taste are mechanoreceptors that allow us to detect textures, like coconut. And there are, believe it or not, pain receptors that contribute to our sense of taste, and that's how we detect things like hot peppers. Which cranial nerves carry taste information? There are three: the facial, glossopharyngeal, and the vagus nerve. Next, we're going to talk about hearing and balance, and again, we'll start with a video.
Both hearing and balance occur within the ear. In hearing, sound waves pass from the outer ear into the auditory canal. They strike the eardrum, making it vibrate. These vibrations travel to the middle ear, setting into motion three tiny bones: the hammer, anvil, and stirrup. The vibrations continue through the cochlea and deep into the inner ear, which contains a fluid reservoir. As fluid ripples across membranes, it stirs tiny hairs connected to nerve endings. The stimulated nerve endings carry impulses into the brain, where they are interpreted as sound. Also located in the inner ear is the equilibrium or balance center. The center sends continuous messages about the body's position to the brain. This information helps us maintain balance.
Okay, so that's a good overview of hearing and balance, both located in the ear. And hearing and balance both use hair cells. And here's what a hair cell looks like. It has these cilia on the top. When the cilia are bent, and they're bent when fluid moves across them, they change the amount of neurotransmitter they release. These hair cells are located in the membranous labyrinth, which is protected by the bony labyrinth. So here's the membranous labyrinth. This part is the cochlea for hearing. Over here we have the semicircular ducts or canals, which are for rotational movements. And inside the vestibule we have the utricle and saccule. We're going to first talk about balance. These are the semicircular canals, and they detect rotation. This is a close-up on the cupula, which is inside the ampulla, and as fluid moves across here, it will bend the cupula, which will bend these hair cells and activate these receptor cells. And so here does it rest, and as the fluid moves, it's going to rotate this, or depending on which way we're rotating, it will rotate it the other way, and that will generate the appropriate action potentials. And the semicircular canals are located in these three different orientations for every possible rotation, every plane. The utricle and the saccule are located here in the vestibule, and they detect gravity and linear acceleration. So they have these little things called otoliths, which are look like crystals, and they're on this gelatinous material, but as you move, for example, if you move forward, because of inertia, these are going to move the opposite direction and bend these hair cells, and then they're going to generate action potentials, and then you're going to sense that you're moving forward or up and down or wherever you're going. And so this shows how they bend in response to gravity, and bend these hair cells, causing depolarization in these cells. Alright, next we're going to talk about hearing, and we have a video that's specific for hearing.
The many sounds in our environment range from the quiet tick of a clock to the roar of a jet engine. Sound is produced by a vibrating object, like a guitar string. These vibrations create a sound wave that moves through the air, traveling outward from the object. When these waves enter our ears, we perceive them as sound. The human ear is composed of three regions: the outer, middle, and inner ears. The outer ear, or pinna, acts as a funnel for conducting air vibrations to the tympanic membrane, or eardrum. The middle ear is a small air-filled space in the skull that contains the ossicles, the three smallest bones in the human body. When vibrations strike the eardrum, the ossicles act as a system of pistons, transmitting the vibrations to a membrane that covers the oval window, which is the opening into the inner ear. The cochlea, which means snail, is a fluid-filled spiral chamber in the inner ear that is about half the size of a garden pea. Running through the center of the cochlea is the organ of Corti, which is the actual receptor organ for the sense of hearing. It contains thousands of hair-like structures called cilia. When the oval window membrane vibrates, the vibration travels through the cochlea and moves the cilia. This fires action potentials that send nerve impulses through the auditory nerve to the brain. This auditory information is transmitted to the primary auditory cortex in the temporal lobe, then to the secondary auditory cortex, and finally to the higher order auditory cortex.
The processing of sounds, ranging from simple tones to more complicated speech perception, becomes increasingly more involved for the brain at each ascending level.
Okay, so that video gives us a good overview of hearing. The sound waves are going to travel through the outer ear, to the middle ear, through the auditory ossicles, and to the cochlea. And so the stapes presses here on the oval window, and let's look closer up at that. Here's the stapes pressing on the oval window. That causes a pressure wave in the perilymph, which is the fluid in the cochlea. And as the basilar membrane moves, these different hair cells are activated. So here's a close-up on the organ of Corti. Here's the basilar membrane. And as it vibrates, it's going to move these hair cells and activate those receptors. So the process of hearing, this shows again an overview of the whole process. Sound arrives at the tympanic membrane where these pressure waves are converted into vibrations, travel through the malleus, incus, and stapes, and then to the cochlea. They are going to distort the basilar membrane and activate hair cells in that region, and then those pressure waves leave through the round window. And as the pressure waves come in, they distort the tympanic membrane, and that moves this and activates these hair cells. The pitch is determined by the location in the cochlea that it excites. So a super high frequency sound is going to excite the basilar membrane right here, medium pitch sound right here, and then a lower frequency sound, the bass tones, will excite it down at this end. So that's how we determine the pitch of sounds. And the volume is determined by the number of hair cells stimulated. So different hair cells have different thresholds. So this one might be activated by a whisper, but this one over here is only activated by a yell. So depending on how loud the noise is, will activate a different number of hair cells. And we can localize sounds because they reach our ears at different times. So for example, a sound on the right side of us is going to reach our right ear before our left ear. And this shows the path from the cochlea. It's going to go here into the medulla oblongata, then into the inferior colliculus where there are reflex centers, then to the thalamus, like all sensory information, and then finally to the cerebral cortex. Next, we're going to talk about vision, and we have a movie for that.
Vision is our dominant sense. 70% of all sensory receptors are found in the eyes, and almost half of the cerebral cortex is involved in vision. The eye contains two kinds of light receptor cells, rods and cones, named for their shapes when seen through a microscope. There are three types of cones, each of which is most sensitive to one of three colors: red, blue, or green. When light enters the eye and activates the cones and rods, they fire action potentials, that send nerve impulses through the optic nerve at the back of the eye. These signals travel to the thalamus, where they synapse on neurons there. The visual information is then sent to the cerebral cortex, where it is processed and relayed to the rest of the brain.
Okay, so the eye is made up of three tunics or layers. And if we look at this figure, we can see the outer layer is called the fibrous tunic, and it's made up of the sclera, which is the white part of the eye. That provides protection. And then also in front we have the cornea, which is clear, and it helps to focus light. Then the middle layer, or the vascular tunic, contains the iris, right here, that controls the diameter of the pupil. Also consists of the ciliary body, which holds the lens in place. And then the choroid, which is the darker layer, and it also contains the capillaries that deliver nutrients to the eye. And then the inner layer, or the neural tunic, consists of the retina, which contains the photoreceptors. And here's just another view of, we can see in front the cornea, the iris, and the lens. And then in the posterior part of the eye is where the retina is. Okay, so we're going to talk about some accessory structures of the eye. The eyelashes, the eyelids, and tears, they all keep the eyes clean. The conjunctiva helps with that too. Here's the lacrimal apparatus, which makes the tears. And the way we see, light passes through the cornea, then the pupil, then the lens, and then to the fovea. The fovea is where you have the highest density of cones, and therefore your sharpest vision. Your pupil, there are two different muscles in the pupil, so it can dilate or constrict in response to the light conditions. In dim light, the dilator muscle is going to contract, and that's going to make your pupil bigger to let in more light. In bright light, the constrictor muscle will contract, and that will close your pupil and protect your retina. Light coming into the eye needs to be focused on the retina. We want our focal point to be right on the retina. And the ciliary muscles, they can change the shape of the lens to focus the light. So for close vision, the ciliary muscles contract and the lens is more round. That focuses the light on the fovea. For more distant vision, those muscles relax and the lens is more football shaped instead of round, and that enables these far light rays to be focused on the fovea. In the process of visual processing, the image reaching our retina is inverted and it's reversed, but our brain is going to straighten this out for us. If we look closer at the retina, we'll see that it's made up of rods, cones, and other neurons. So the rods are rod-shaped, and you can't really tell the difference from this picture. This is an actual photograph of the retina. And you can see this darker spot is the fovea, and around that is the macula lutea. Here's the optic disc. We can see blood vessels radiating from that. That's also where the optic nerve is going to be formed. So here's a close-up on the retina, and we can see the rod-shaped ones are rods and the cone-shaped ones are cones. The cones are very sensitive and they allow us to see in dim light, but they cannot distinguish colors. That's why when you go out at night, you only see in black and white. Cones provide sharper, clearer images and color, but they require more intense light to be activated. And the rods and cones send signals through bipolar cells. These are the bipolar cells right here. And then the retinal ganglion cells. This figure shows our visual acuity and also our density. The purple is our density of cones. You can see the density of cones is highest here in the fovea. And then the density of rods is down here. Density of rods is highest in our peripheral vision. That's why our peripheral vision is not as sharp, because we don't have any cones there, or very few cones. And our visual acuity is highest right here in the fovea, and that's where we have the highest density of cones. So a little bit how we actually see. Light comes into these, and if we look closer, we'll see all these discs in the rods. And if we look even closer at one of those discs, we'll see that it contains rhodopsin. So rods and cones contain membranous discs, which we just saw here. Here's the discs. And they contain plasma membranes, which contain the visual pigment, which is rhodopsin. And when light strikes this visual pigment, this molecule here, retinol, it changes its shape. So here's a photon of light striking it. It changes its shape. It goes from bent to straight. This leads to ion channels closing. So now sodium can no longer enter. This changes the amount of neurotransmitter the cell is releasing. This is going to lead to action potentials in the retinal ganglion cells, which are going to become the optic nerve. ATP will reactivate the retinol, so it's bent again, and then it's ready to absorb another photon of light and activate these cells again. Rods and cones are a little bit weird in that they're actually releasing neurotransmitters in the dark, but they're releasing inhibitory neurotransmitters. So they send an inhibitory neurotransmitter to the bipolar cell, and we don't get a signal in the ganglion cell. When light hits them, they stop releasing this inhibitory neurotransmitter, and then we get action potentials in the bipolar cells, and that leads to action potentials in the ganglion cells, and it gets interpreted as light. We have 3 types of cones, and each one is specific for a different color of light. There are blue cones, green cones, and red cones. And our rods are kind of in this green range. That's what they're most sensitive to, but they don't really distinguish between colors.